DNA damage is the root of aging. Genotoxic stress drives cellular senescence, systemic inflammation, and the collapse of human health span. As DNA damage accumulates, the structure unravels, leading to double-strand breaks and ultimately systemic collapse. The evolutionary paradox of aging, known as antagonistic pleiotropy, highlights the biological cost of avoiding cancer early in life. Aggressive DNA damage response suppresses tumors in youth, but leads to the accumulation of senescent cells and tissue degeneration later in life. Our genetic blueprint is under a relentless siege. Endogenous threats like depurination and single-strand breaks occur thousands of times daily, outpacing exogenous threats like radiation by orders of magnitude. The majority of damage comes from within, through normal cellular metabolism. The cell has a defense arsenal against DNA damage, including base excision repair, nucleotide excision repair, non-homologous end joining, and interstrand crosslink repair. These pathways correct small lesions, remove bulky adducts, ligate broken strands, and resolve toxic crosslinks. Unrepaired DNA lesions force a choice at the crossroads of cell fate. After detection and amplification of the signal, the cell decides between transient arrest for successful repair and homeostasis, cell death via apoptosis, or irreversible damage leading to senescence. A senescent cell is actively repressed to prevent the replication of a mutated genome. Key features include arrested replication, structural degradation like reduced lamin B1 expression, epigenetic alterations that lock down the genome, and the expression of biomarkers like SA beta-gal. Senescent cells drive systemic chronic sterile inflammation, creating an inflammatory contagion radius. Factors like NFKB activation and SASP degrade local tissue architecture, accelerating the aging of surrounding healthy tissue. Unrepaired DNA damage is the unifying root cause of aging. Genotoxic stress perturbs cellular homeostasis, leading to chronic inflammation, mitochondrial dysfunction, altered proteostasis, cellular senescence, and epigenetic drift. The architecture of accelerated aging is evident in progeroid syndromes, where mutations in DNA repair or architectural genes lead to premature aging. Examples include Hutchinson-Gilford progeria and Werner syndrome, which involve defects in chromatin organization and replication, respectively. Accelerated aging can stem from structural defects, like in progeria, or repair defects, as seen in Werner syndrome. Whether the genome breaks from architectural collapse or biochemical failure, the result is identical: accelerated aging. Iatrogenic aging, the cost of surviving cancer, demonstrates how DNA-damaging treatments can accelerate biological aging. Chemotherapy can cause a spike in P16INK4, expression equivalent to 15 years of natural chronological aging, matching frailty indices and disease onset rates of a much older person. Measuring the unseen biological age requires multi-endpoint tracking. Key markers include P16INK4A expression, the most reliable transcriptomic marker for biological aging, and SA beta-gal activity, the defining enzymatic signature of senescent cells. The NAD plus sync creates a metabolic vicious cycle. Persistent DNA strand breaks activate PARP1, which consumes NAD+. This continuous demand depletes the cell's energy currency, shutting down longevity networks like sirtuins and causing mitochondrial collapse. We can tip the scales toward longevity through interventions like caloric restriction, NAD+ replenishment, and senolytics. These strategies mitigate DNA-damaging species, restore mitochondrial homeostasis, and clear SASP-secreting senescent cells. Aging is a highly ordered, evolutionary compromise triggered by the relentless siege on our genetic blueprint. The lifespan blueprint progresses from rapid chemistry damage, through signaling and fate selection, to cellular contagion, eventually resulting in systemic pathology and the failure of the human machine. Thank you for watching this video on the genomic root of aging. We hope you've gained a deeper understanding of how DNA damage drives the aging process and the potential for interventions to extend health span.